So good morning. My name's Andrew Zaleski, and I'll be chairing this morning's session on brain heterogeneity. It's a great pleasure to welcome you all here to our session. So I'd first like to thank Dr. Junhao Wen, who unfortunately hasn't been able to join us, but he's done all of the heavy lifting in organising this morning's symposium. And he will give a pre-recorded talk. He's the last speaker of our session. If you have any questions, there is a microphone here at the front. And of course, you can type in your questions on the app, and I'll endeavour to read those out. That said, we've got a fairly jam-packed session. We've got five speakers this morning, so I think we'll only probably have time for one or two questions after each session, and if we have time at the end, we can also have a Q&A session. So without further ado, I'd like to introduce our first speaker, Dr. Maria Di Biase from the University of Melbourne, who will speak on heterogeneity in psychiatric disorders. So over to you, Maria.
Thanks, Andrew. Sorry, hopefully you can hear me. It's always a challenge when you're short. So I guess one of the reasons why we're here today is because psychiatric disorders are often described as highly heterogeneous, especially as compared to non-psychiatric illnesses. And this presents a challenge for scientific progress and for clinical trials. But what do we really mean when we describe the disorders this way? I guess the degree of biological variability across and within psychiatric disorders and even across non-psychiatric disorders is not fully understood. And heterogeneity is a relative concept, so are we saying that psychiatric disorders are more heterogeneous than, say, neurological disorders or autoimmune conditions? And if so, by what measure? So I think these questions are really important because we often treat heterogeneity as a major barrier to research. So understanding the scope of heterogeneity, both within psychiatry and beyond, will help us to understand where the challenges might lie and especially how we can leverage this information moving forward. So here in recent unpublished work, we've begun to address this question, just how heterogeneous are psychiatric disorders. We've considered a range of biological phenotypes, including 60 biochemical phenotypes, which were grouped into 11 panels, and 5 brain MRI phenotypes, and these were primarily acquired through the UK Biobank. For each phenotype, we established normative ranges of variation, so reference charts as a function of age and sex in over 50,000 healthy individuals. And then we computed how individuals deviate from this normative range for individuals with a particular illness. Heterogeneity was then quantified for 9 psychiatric and 30 non-psychiatric illnesses, and this was simply defined as the spread or the standard deviation across the deviation scores within an illness category. So here you see illnesses ranked by heterogeneity, which is on the y-axis. The teal colour shows the average of the biochemical traits, and the brown colour shows the average of the brain traits. And what you can see is that all of these illnesses are more heterogeneous than healthy normative variation. So a heterogeneity index of greater than 1 is greater than healthy normative variation, at least for the biochemical traits. And then most illnesses are also more heterogeneous across the brain traits. You can also see that in general, biochemical traits are more heterogeneous than the brain traits. However, for brain-related illnesses such as schizophrenia, multiple sclerosis, epilepsy, stroke, the reverse holds true. Sorry, I'm just trying to click here. Yeah. So of the psychiatric disorders, schizophrenia exhibited the greatest heterogeneity across both biochemical and brain domains, showing up to a 20% increase in variability compared to healthy individuals. And this was followed closely by bipolar disorder. In contrast to our expectation, psychiatric disorders did not show systematically or significantly increased biological variability relative to broad groups of other illnesses, including neurological, cardiovascular, gastrointestinal, musculoskeletal, and respiratory illness. So here we challenge the assumption that heterogeneity is uniquely found in psychiatric disorders. Instead, what our findings suggest is that this is a broader pattern of chronic illness. Another finding here was that the traits exhibiting the greatest heterogeneity weren't necessarily the same as those showing the largest mean or case-control difference. For example, in multiple sclerosis, subcortical brain volume emerged as the most heterogeneous trait. Now, this is consistent with evidence pointing to this marker as an established predictor of disability progression in the disorder. In contrast, we found that white matter anisotropy showed the largest between-group difference, which makes sense, given that white matter is the primary tissue affected in this illness. So in this way, we often found that mean group differences highlighted primary features of illness, while measures of heterogeneity, they expose dimensions along which individuals differ within a diagnostic category, offering potential to identify biological subtypes and also prognostic markers of illness. Now, this became more interesting when we considered psychiatric disorders where we don't have clear biological markers. For example, in schizophrenia, surface area was the most heterogeneous trait. So this is interesting because surface area develops over a long period of around 10 years in childhood. There's a lot of time for variation to ensue and for environmental factors to impart increased vulnerability to illness and to structural changes in the brain. In contrast, the largest mean group difference was in cortical thickness. So this is a more universally affected feature in the illness, which peaks in early brain development. So this is consistent with epidemiological and genetic data that suggests that early neurodevelopmental mechanisms are important in this disorder. So here, critically, we're pointing to two different potential intervention windows for the same diagnostic group, which is why it's critical not just to capture mean group differences, but also heterogeneity. We next asked whether genetic factors contribute to illness-related heterogeneity. And to do this, we quantified polygenic scores for each biological trait, and this is important to emphasize here. These aren't polygenic risk scores for an illness. These are genetically predicted scores for the particular biological traits. And then we regressed out these genetic effects before recomputing heterogeneity. So the brown bars here show heterogeneity before polygenic score adjustment and the yellow bars after polygenic score adjustment. And as you can see across the illnesses, accounting for polygenic scores decreased heterogeneity only very modestly by less than 5%. Also, the illness rankings remained broadly consistent before and after controlling for polygenic scores. As expected, these genetic effects did account for more heterogeneity in some traits relative to others, such as cortical thickness relative to surface area. But the key message here is that polygenic architecture of the biological phenotypes accounts for only a small portion of illness-related biological heterogeneity, and the rest likely reflects other genetic factors, including illness-related genetic factors, environmental factors, and gene-environment interactions. So here, we developed a disease hierarchy based on heterogeneity, allowing us to understand, relatively speaking, just how heterogeneous these disorders are, including many psychiatric conditions. And we hope that this work will serve as a resource of biological markers that are heterogeneous, that can be used in future work to help guide predictive models of treatment response, prognostic stratification, and disease mechanism-based treatment development. So where are we taking this to next? Instead of seeing or viewing heterogeneity as a challenge or an issue that we need to solve, we're utilising it as a source of insight. So over the last 5 years, I've led the establishment of the Australian Psychiatry Stem Cell Resource. We've generated many stem cell lines from individuals at the earliest stages of psychosis, and here we have matched genotype, longitudinal neuroimaging data, and clinical profiles. So just to briefly describe this process, we take somatic cells, skin or blood, and then reprogram these to become induced pluripotent stem cells, which are capable of becoming various brain and body cell types, and we're coaxing them to become brain cells that we can study in vitro. So why use these induced pluripotent stem cells? As I just mentioned, we can generate diverse brain cell types. We can study disease mechanisms and screen for drugs safely because these are experimental models in a dish. Importantly, we can personalize research and therapy because these cells that we end up with retain the genetic background of the donor from whom they were derived. giving us a fantastic opportunity to start addressing those questions around heterogeneity. And because they do this, we can study how features of brain development may have looked in these individuals without directly accessing their brain tissues. So this is a really powerful platform that allows us to study dynamic disease mechanisms at a cellular and molecular scale. So we are transforming or differentiating these stem cells into cortical organoids, which are 3D cellular models of the brain, and this is work driven by PhD candidate Jordan Clark. And we're doing this with developed methods to do this at scale, so we can look at these organoids in many patients with sufficient sample sizes. By tracking their gross morphology across time, we can compare variability in their growth trajectories between patients and healthy controls. For example, from day 40 to day 60, we see this rapid increase in volume reflective of cell proliferation, and then a less pronounced increase from day 60 to day 80 as they transition from proliferative to post-mitotic stages. We're also testing the morphology of these rosette-like structures, which reflect early neuroepithelial organization and cortical development. By day 80, our cortical organoids start to exhibit early features of organized laminar architecture, with upper layer Reland marker concentrated to the outer surface, and with specificity of projection markers, such as can be seen with SAPB2, an upper layer marker, CTIP2, a deeper layer marker, and TBR1, a deep layer marker. Older organoids also exhibit features that are more mature. For example, we lose these rosette-like structures and see more uniform expression of class 3 beta tubulin, a post-mitotic neuron marker. Now, these layer-specific markers, by measuring their width, provide a direct estimate of cortical thickness in vitro. And what we're doing is measuring cell type specific features and transcriptional signatures that might underpin cortical thickness deficits that we observe in vivo with MRI. Of course, connectivity is another major or potentially major driver of heterogeneity of brain structure and function that we observe with MRI. So in this work driven by PhD candidate Cassandra Hoffman, we're developing and mapping measures of structural network connectivity in vitro as a more direct cellular correlate of what we observe in vivo with diffusion-weighted imaging. By connecting what we're seeing at the microscale, cellular organization, and network level graph theoretical differences, and then mapping these to systems level differences that we observe in the brain, we can begin to uncover individual mechanisms, that is, how individual variability at a cellular and molecular scale contribute to the macro scale biological heterogeneity that we observe in the brain through MRI. So just some key takeaways here. Psychiatric disorders are not necessarily uniquely heterogeneous. In fact, we see that heterogeneity is comparable to that of many non-psychiatric illnesses when we consider a broad panel of biological markers. Also, while the field is commonly focused on mean group differences, this isn't the whole story, and I think this is becoming very apparent with the applications of normative modelling that we've been seeing. We found that biological heterogeneity offers insights into disease biology, potentially revealing developmental or trajectory-based subtypes. And we're leveraging multimodal and multi-scale approaches to help bridge the macro and micro-level data to uncover mechanistic and developmental roots of heterogeneity in psychiatry. So while MRI provides a snapshot, we want to provide the movie, the dynamic process of how we get there. I'd like to gratefully acknowledge the participants for volunteering their time in these presented studies, Andrew for organising this symposium, and all the collaborators involved in this work. So thank you for listening, everyone.
Thanks very much, Maria, for the fantastic overview of heterogeneity across scales, all the way from imaging to neuronal levels. Perhaps we can take one quick question from the audience. There's a microphone at the centre there if you wanted to.
Okay, so at least in healthy subjects, typically heterogeneity between brain structure, cortical thickness, myelination, and so on, is bigger between brain regions than it is at a given brain region between individuals. So wondering, when you have these organoid samples, what are they meant to represent, a specific brain region, or are they supposed to just be somehow representative of the individual's brain in a broad sense?
Yeah, this is a really important question. So in terms of deriving organoids, we can use unpatented or patented approaches to develop more specific brain regions. So we're using 4 brain patented protocols so that we've got more specificity in terms of what regions of the brain we're representing in these organoids. But this won't be representative of ventral regions, subcortical regions. So this is something that we need to really consider in organoid research and how we can model the differences between brain regions. So thank you for that important question.
Okay.